Imagine a microscopic engine that turns thousands of times per second, powered not by fuel but by a tiny electrical charge across a cell membrane. In this lesson we uncover how bacteria use the flow of protons to spin their flagella and move through their world. The bacterial flagellum is a complex nanomachine that propels cells through liquid environments. Its outermost component, the filament, extends several micrometers and acts like a helical propeller. Connecting the filament to the cell body is a short curved structure called the hook, which functions as a universal joint. The motor core resides within the cytoplasmic membrane and comprises a rotor ring surrounded by stator units, anchored in the wall. Stator units form ion channels that conduct protons from the periplasmic space into the cytoplasm. As protons flow through these channels, they generate torque on the rotor via electrostatic interactions. The generated torque rotates the rotor, which is mechanically coupled to the hook and filament. This rotation translates into forward or backward swimming depending on the direction of proton flux. The entire system operates with remarkable efficiency, converting a chemical gradient into mechanical work. Structural studies reveal that the rotor contains multiple copies of the protein Flag, essential for torque generation. Mutations in stator proteins disrupt proton flow and abolish motility, underscoring their critical role. Thus, bacterial flagellar rotation exemplifies how a proton motive force can power a rotary motor at the nanoscale. Pause 0.8. The bacterial flagellum is powered by a proton motive force generated across the cytoplasmic membrane. Protons accumulate outside the cell, creating both a chemical gradient I circumflex pH and an electrical potential I circumflex I umlaut letter QC6. The I circumflex pH component reflects a higher proton concentration in the periplasm compared to the cytoplasm. The I circumflex I umlaut letter QC6 component represents a negative charge inside the cytoplasm relative to the exterior. Together these gradients form an electrochemical potential that can be quantified as I circumflex P equals I circumflex I umlaut letter QC6 A circumflex euros 2.303 RT/F I circumflex pH. When protons flow back into the cell through specialized channels, they release stored energy. This proton flux drives conformational changes in the motor euros trade S stator units embedded in the inner membrane. The stators convert the chemical energy of the proton gradient into mechanical torque applied to the rotor. The rotor, connected to the filament, rapidly rotates at speeds exceeding 10,000 revolutions per minute under highly optimal conditions. Experimental measurements show that blocking proton flow halts rotation, confirming the dependence on I circumflex P. Thus, the flagellar motor exemplifies how a biological system harnesses an electrochemical gradient for mechanical work. Pause 0.8. The stator complex is composed of 4 copies each of MotA and MotB proteins that embed in the bacterial inner membrane. MotA contains 6 transmembrane helices, while MotB contributes a single helix that anchors the complex to peptidoglycan. Together they form a continuous proton channel whose narrow pore aligns with a rotor ring at the base of the flagellum. When the proton motive force drives protons from the periplasmic side into this channel, electrostatic interactions between charged residues in MotA and MotB create a local electric field. This field induces conformational changes that rotate the MotA helices relative to MotB, effectively converting ion flow into mechanical torque. The rotation of the MotA helices is transmitted through the stator rotor interface to the C-ring, causing the rotor to turn in a clockwise or counterclockwise direction depending on proton flux direction. Experimental mutagenesis of key acidic residues in MotB demonstrates that loss of charge abolishes torque generation, while preserving ion conductance. Cryo-EM reconstructions reveal that the channel lumen is lined with alternating hydrophobic and polar side chains, guiding protons via a Gruthos-like hopping mechanism. The coupling efficiency of this system is remarkably high. Each proton translocation can generate several piconewton-meters of torque sufficient to propel the flagellum at speeds exceeding 10,000 revolutions per minute. Thus, the MotA/MotB stator functions as a molecular machine that directly links the electrochemical gradient to rotational motion without intermediary energy storage. Pause 0.8. The flagellar motor converts the electrochemical gradient of protons into mechanical torque. Protons enter the periplasmic space and bind to acidic residues on the MotB subunit of the stator complex. Binding triggers a local rearrangement that pulls the adjacent MotA transmembrane helices toward the membrane core. This movement shortens the distance between MotA and the rotor euros trade SC ring, creating an electrostatic push on the Flag protein. The resulting force rotates the C-ring counterclockwise when viewed from the cytoplasmic side, initiating shaft rotation. As the ring turns, the stator remains stationary, repeatedly cycling proton binding and release to sustain continuous motion. Each proton cycle generates a small torque increment. Cumulative increments produce smooth, high-speed rotation. The motor Euro's efficiency depends on the density of functional MotA/MotB units embedded in the cell membrane. Mutations that disrupt proton binding sites abolish rotation, confirming the direct link between proton motive force and torque. Thus, the stator acts as a molecular transducer, converting chemical energy into mechanical work via conformational changes. Pause 0.8. The flagellar motor converts a proton gradient into mechanical work by coupling ion flux to torque generation. Protons enter through stator channels, creating an electrochemical potential that drives rotation of the rotor ring. Each proton translocation event releases energy equivalent to a small angular displacement of the rotor. The cumulative effect of many such events produces a continuous torque that pushes the flagellum against viscous drag. When load increases, more protons are required per revolution, slowing the motor but maintaining torque output. Conversely, under low load the same proton flux yields higher angular velocity, because less energy is dissipated as work. The relationship between ion flow rate and generated torque can be plotted as a characteristic torque Euro speed curve. On this graph, the higher Euro's load regime appears near the plateau where torque remains constant while speed drops sharply. In the lower Euro's load region, the slope of the curve steepens, indicating that small increases in proton flux translate into large speed gains. This balance between ion motive force and mechanical load ensures efficient propulsion across diverse environmental viscosities. Pause 0.8. The proton motive force is transduced into mechanical work by the flagellar motor through a series of well-defined biochemical steps. Early experiments used bead assays to attach micron-sized beads to the hook, allowing observation of rotation under a microscope. Optical tweezers later refined this approach by applying calibrated forces and measuring displacement with nanometer precision. In these setups, a laser trap holds a polystyrene bead while the motor turns, generating measurable torque that can be quantified in piconewtons per radian. Fluorescence microscopy of stator proteins labeled with green fluorescent protein revealed their dynamic assembly at the motor during active rotation. Time-lapse imaging showed that stators bind and unbind on a millisecond timescale, correlating with changes in torque output. Single-cell torque measurements using magnetic tweezers demonstrated that each proton translocation contributes a discrete step of about 0.1 pNa circumflex nm to the total torque. These data were plotted against membrane potential, revealing a linear relationship that matches predictions from the proton-driven model. Cryo-electron tomography provided high-resolution snapshots of motor architecture, confirming the spatial arrangement of rotor and stator subunits required for efficient energy conversion. Mutational analysis of key residues in the MotA/MotB proteins further validated the role of ion channels in driving rotation. Together, these experimental techniques converge to support a mechanistic framework where proton flow directly powers flagellar motion. The consistency across diverse measurement modalities strengthens confidence that the proton motive force is the primary energy source for bacterial locomotion. Pause 0.8. We reviewed the architecture of the flagellar motor, the electrochemical basis of the proton motive force, how stator complexes form ion channels that translate proton flux into mechanical work, and the experimental evidence supporting this model. Understanding these mechanisms illuminates how bacteria convert chemical energy into motion.